Our next speaker is Dr. Dennis Bashaw. He's a senior science advisor at the FDA, and he will be continuing our discussions on sunscreen products. And his presentation is IVPT studies with sunscreen products, potential regulatory utility.
I'm here to speak this morning on a topic that really is going to build on the fine presentations this morning from Dr. Maibach, Ronnie, and Dr. Yang, but from a different perspective. I'm speaking now, I've taken off my scientist hat and put on my regulator hat, but one thing we do need to clarify, I have left the FDA after 34 years, decided to leave the FDA, and just like that, I have a long disclaimer here. Basically, I am not speaking for the FDA. I'm not representing the FDA. I'm not here presenting FDA policy, not speaking for on behalf of the FDA. I got to make that very clear because obviously after 34 years of association with the FDA, one always makes that assumption. And the last thing I want to have happen is someone hears something I say here, and then they go to the FDA for a meeting and say, well, Dr. Bashaw said, as if I spoke on behalf of the FDA, but I am no longer at the FDA. But I do have a lot of experience in this area and want to share a perspective that a lot of people don't really think about when they're doing these studies and looking at them and the opportunities we have facing right now. Disclosure at this point, I don't have any disclosure, so I just want to make that clear. At this point, I'm speaking totally without any encumbrance right now. And a general outline right here for the talk, because of the time limitations. My slides are over detailed. I am not going to read them to you as they're going to be made available to you as a reference, but I will refer to the different high points of them to get the thesis moving here. And the real first question is regulatory utility, something that most scientists never think about. We think about doing a project, we think about doing a paper, we think about answering a problem, but then the next question is about the regulatory utility of it is something that doesn't really come to mind. So I'm going to ask you all today to please think like a regulator, put on your regulator hats, because the FDA and the Center for Drug Evaluation and Research does have a dual role to balance the needs of regulations, to have a regulated industry that's well ordered and we have a high confidence in the answers, and one where science is promoted and we promote innovation. The FDA certainly has a role there, and I think that both Dr. Ronnie and Dr. Yang have showed today in their presentations the depth and the variety of the facilities and the research being conducted and how it can move the science forward in a very positive way. Because, well, people do, like I always tell a regulator, they always quote regulations. And I think the first thing we've got to accept here is that the regulations clearly allow for the use of appropriate in vitro tests in the establishment, in the regulation of bioavailability and bioequivalence. In vitro tests are allowed. They just have to meet certain criteria. They have to mean something. They have to be related back to clinical utility or some other aspect that we can mark here. And you know, that's been the failing we've had so far, I think, is that we do these tests and we get these results, but we don't think about what's the next step. How do we make them, move them? You know, we always talked about originally in clinical pharmacy, moving from bench to bedside. Well, how do we move from bench to regulation? How do we move the science forward from it just being something we do to something that has regulatory import? Because the regulations do allow for it, and it's taking just time for the science to get up to us. And one of the things that you've heard, again, I'm just going to touch on quickly here, is we've got to have reproducibility and validation across study sites. We can have all the fine methods we want, but if we can't have confidence in their reproducibility, we can't have confidence in their sensitivity, then they're really not of any use from a regulatory perspective. Because again, the way you can use in vitro testing is it has to be relevant clinically. It has to tie back somehow to either clinical safety data, clinical efficacy data. It's got to tie back to that initial approvability package. So I guess the first question is, well, why hasn't, we've been talking, as Dr. Maibach talked this morning, 80 years, why hasn't IVPT moved into regulatory use? The science is generally well established. We all agree that. We have commercial single and multi-cell devices. we use it in formulation. And the real question, I think, has been what's lacking? Standardization of methods and reproducibility. As I said, we don't want to repeat the DPK experience where we had a guidance put out by the FDA using DPK and it had to be withdrawn because there were reproducibility issues between different sites. We need to have an identified need too. In other words, you can have a test, but it can't be a method in search of a problem. The method has to address a problem, has to address a need. And I think that we have that opportunity now, and Dr. Yang has talked about it, and that opportunity is sunscreens. I think sunscreens present us with the best case for making IVPT really come to light in the regulatory role. As we already talked about, the question whether or not sunscreens are absorbed has been answered. I mean, the FDA, both myself, Dr. Yang, and others were on these papers. I think we all agree now that although originally I heard many people tell me that sunscreens were not absorbed because they weren't formulated to be absorbed, well, as Dr. Yang and others have pointed out, and these studies have clearly shown they are absorbed. Their formulation is such they do get absorbed. And I think that if we think about from the IVPT perspective, it's not just that they're absorbed in a time frame that works with IVPT. I mean, we can see just looking at one set of data from the most recent FDA MUST study, we can see that we get absorption detected in the plasma within a few hours. Very quickly, you're seeing it, so we know we are transiting the skin fairly well, fairly rapidly. And we are producing levels there well in the ranges of some issue of concern. I mean, we have to get some safety data on. So we know it's absorbed. We know it's absorbed relatively quickly. It's not like it takes a long time to get levels. It's transiting the skin relatively quickly. And this all builds on a way for making IVPT, again, tie back clinically. We saw from Dr. Yang's research that, again, we're seeing it in the systems very quickly as well. So we do have a sort of a linkage there. If you want to think back to some of the USP correlation levels, A for IV, IVC, A, B, and C, we sort of have the C-level correlation already there. We're seeing that linkage of, initial linkage of absorption and permeation working together here. And I think that's why it's really the vehicle, sunscreens can be the vehicle for making IVPT a more recognized in vitro test at the FDA. And I think it's primarily because the second issue right there, sunscreens undergo significant reformulations. The sunscreen products, the copper tone or the banana boat that you're buying today is not what you were buying 10 years ago. The formulations have been changed. They are continually tweaked. To make them drier, to make them more cosmetically elegant, to make them more deployable on the skin. These formulations continually change. Now, we've shown with the maximum usage trial, which of course I am a proponent of, there's no two ways of saying it, that it does show absorption. But a MUST trial is not something you want to use for routine testing all the time, not for formulation testing. An IVPT approach would be much more cost effective, much faster, and would get us an answer we could make actionable on. So I think that that's where, you've got a method, you have a need for it, here we go. We can now use this with the sunscreens to push IVPT forward, much as the way Dr. Ronnie with his Q3 issue development with acyclovir has helped us by moving these methods more to the forefront. And I think you can't lose sight of the cost of trials, because it's easy to say, go do a trial. I just spent $1 million. It's very easy to do that. You've got to look at where you're at on this continuum for informational value versus conduct. And also cost. Cost always drives things. You really can't get away from that. As much as we'd like to think about it, we do have to address it here. We do have to recognize it. And we start thinking about the quality of studies we're normally at. IVRT, IVPT study is relatively easy to do compared to a clinical trial. Its information value, when it's properly validated, can be very high. It's a kind of thing that we need to take the investment into, because yes, while we can continue to do MUST studies for these kinds of changes are really not a good use of resources. And of course, while its informational value is high, it's not especially difficult to do, the cost is there. I neglected to show I did it for a height effect, that the IVPT, IVRT doesn't cost as much as a must. That's an oversight on my slide there. I should have made that bar shorter. But the point being here is that it still can give us actionable answers. And I really believe that this is the way to move it forward. So we know it's a key element in formulation selection right now. Companies are doing IVPT to pick one formulation versus another for various reasons. Dr. Ronnie is a Q3 we already mentioned about today. And the FDA has been putting out last year we put out 2 articles, one by my group at the FDA when I was still in the FDA, and one by Dr. Young's group, talking about best practices to how we can get an IVPT methodology that can be standardized, that we need to have it be reproducible. One of the failings I feel that we've had throughout the years is that everybody likes to do it their way. Everybody likes the way their laboratory prepares samples. Everybody likes the way their group does this. And that is holding it back, because what we need to have is a methodology that's consistent, that we can look across products, that we can say how these things relate to each other, and how we can look at formulations, because you don't want to have gone to one contractor and they do an IVPT one way, then you go to a second contractor and they do it slightly differently. Because that was what was the DPK experience, is there were slight differences in the methodology used at the contractor site. So if we were to think about, from a regulatory standpoint, still our regular hats on, what's our acceptance scorecard? Where are we at here? What do we have? We have a defined need to assess formulation changes in an in vitro setting that can maintain preclinical safety information. That's one of the things we're really concerned about, is preclinical safety, making sure that information stays relevant. We have a methodology that's probably as close to the system as we're going to get. Sans microdialysis, which I'm a big proponent of also, but microdialysis is not quite to that stage yet, in my opinion. Maybe it will come up in time, but there's more encumbrances with microdialysis. I certainly understand that. And we certainly have both a demonstrated regulatory need, and as you've seen the sponsorship of this workshop, you've heard the speeches previously by previous collaborators. You've seen in publications, the FDA is very interested in the methodology. There's an interest there that this academia, that science and industry can really harness to move forward. But what is missing? Standardization of methods. Again, proliferation, just because you like doing it one way doesn't mean you can do it we've got to have standardization of methods. We've got to have validation of methods. We've got to have within lab and cross lab validation to show and give confidence to the regulator that when they see these results, they can accept them as being specific, reproducible, all the characteristics of a good assay. Because if not, then it just becomes a random, and randomness will kill the methodology quickly. So I know we are short on time. So now we're going to try to wrap up, because again, I'm trying to present a regulator's perspective based on my experience while I was at the FDA. So, the past year with me. So where are we going with it? We have a methodology, but we've not used it to its full potential because, quite frankly, a lack of a driving force. This is our fixed law in effect right here. We haven't had a driving force to push it. We now have a real need with the sunscreens, because again, oxybenzone is in literally thousands of products. Avibenzone, the UVA blocker, we've got to do these things because we've got to understand their absorption profile and their safety profile, because there are concerns in the literature about them. So again, we can do must trials all day long if you wish. That is certainly a way to do. It's very expensive and very time consuming, and probably is going to give you more information than you need. And it makes it a large regulatory burden as well on a regulator. So if we can come together and get a methodology, we can really have a win-win for both sides, and it needs to go that way. I think what holds us back is inertia in the system. FDA is a bureaucracy. I'm not going to kid you. There's inertia there. But there's also fear of the unknown from the sponsor side, because, they don't know they want to know what the answer is. And there's always fear when you try to put something new in place, and fear eats the soul. It truly does. So when I was at the FDA, I would get asked these questions. When will the FDA accept this methodology? And the answer always comes back to, when will you do it? Because we can't approve a methodology that the industry and academia hasn't researched and brought and shown us that it has value. We can't just blindly approve these things. And this is what we get, this standoff continuously. There has to be a need. Science and policy does not exist in a vacuum, and we all have a role to play. And I truly believe that there's an opportunity now to use the vehicle, so to speak, of sunscreens to drive the approvability and the acceptability of IVPT testing at the FDA. I believe that there's a defined need. It's not a theoretical need. It's not some kind of future need. It's a now need if we're going to move these process forward. Again, we need to work together. These workshops are useful. They're very valuable to discuss the tools, how they can be standardized, how we communicate, how regulators can participate in the meetings and communicate expectations to stakeholders, and how we can work together to develop actionable information, because good science makes good policy. And then going back to Beal and Shiner, we also need to use a learn and confirm method, where as we do this together, we learn what methods and work procedures, whether it be, dermatomed or heat separated epidermis, we learn and we feed back in and we continually refine the methods. We're doing that, but we've got to come together and we've got to have support from everybody. We've got to have the academia support. We have to have industry support. We've got to have the regulatory support. The regulators, I can tell based on my experience, just speaking for myself as having been there is a desire to have these methods. There's a desire to bring this forward. But again, the FDA, sorry, not we, past tense, the FDA cannot approve a methodology for use without a significant database beneath it to back up the use of it, because you're lowering, some people say, well, you're lowering standards. No, we're not. We have to be able to show that we have maintained the standards. And by doing so with enough panache and enough data to support it. Now, I'm bringing this slide back up again for one reason. I forgot to mention, to remember the slide. I want you to remember, I had this slide much earlier in my presentation. But not only did I have this slide earlier in this presentation, we had it in 2013 at the PQRI. We had it in 2008 at Bio International. And I also had this slide in 2005 at AAPS annual meeting. We have had this discussion year after year after year about developing topical bioavailability data, about using IVPT, about using other methodologies. We have discussed this, at least in my career, I mean, at least since the 2000s, early 2000s, because when I first got involved in dermatology, I'm sure as Dr. Maybach talked about, 80 years it's been out there. We've got to move these things. We've got to quit talking about it, people. We need to start doing the necessary groundwork to get the test to where it can be validated, standardized, and put in place. Now, if 2021 is not IVPT's time, if not, then when? Abraham Lincoln has supposedly said that if he had 5 minutes to chop down a tree, I spend the first 3 minutes sharpening my axe. I pose to you, ladies and gentlemen attending today, that our axe is sharp. We have been sharpening the axe. It's getting to the point now we need to start swinging. We need to start letting the chips fly and getting in there and doing the kind of basic work and build on it, that we can come and say, hey, we think we have a methodology, and here are the bullet points. Here is how we can do it. We can standardize on this, and we can fulfill a regulatory need that can both lessen the burden on industry, lessen the burden on the regulators as well. Again, I do not want to be in this situation where in 5 years from now, in 2026, we're having another meeting on this topic. And we're still having these discussions about, well, is IVPT ready for prime? It's ready for prime time, I fully believe, ladies and gentlemen. But we need to have the case. And I think that Sunscreens give us a case that it could fill the need very clearly. I think that it can move it forward. And it can be the vehicle for change, if you will. I want to thank you all for your time today. I try to keep to my 15. I think I just about hit my time spot online. You see, I've got my kite flying. Since I'm no longer at the FDA, I'm flying free now. So I have my contact information like Dr. Myback. I'd like to thank for those people who've laid the groundwork for me so far today, Dr. Yang and Dr. Myback, and of course, Dr. Ronnie. And for the afternoon speakers, I'm certainly looking forward to it as well, as I have just sort of given you your marching orders. Now let's get moving on this. Thank you very much.